I want us to record the whole thing like that guy that always does the movie trailers. In a world,
Welcome to Absolute Genius.
That's it. Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Cassie McCreary.
And I'm Jordan Ruggieri, and today we are stoked to get our assay on with two of Thermo Fisher's own resident geniuses, Dr. Rounak Feigelman and Kimi Soohoo Ong.
Rounak is a staff bioinformatics scientist at Thermo Fisher with a diverse background in next-generation sequencing, computational biology, and bioinformatics. Kimi works with Rounak in qPCR and dPCR digital assay design. She has experience in pharmacological chemistry and holds a graduate certificate in data mining and analytics. They're both extremely knowledgeable about digital PCR and assay design, and we had a great time learning about the mix of analytics and creativity that they bring to their work. We hope you enjoy our conversation.
Well, Kimi and Rounak, thank you so much for joining us today on Absolute Genius. We are very excited to have you. I'm really excited to dive into the world of digital PCR assays. I think it's a really cool conversation, and we absolutely love digital PCR here at Absolute Genius. So, one question I want to kind of pose to both of you is, when you're looking at digital PCR and developing an assay for digital PCR, what are some of the key factors that researchers should consider?
Some of the things that we try to learn beforehand when we start designing an assay is what's the kind of sample that we are dealing with. So what's the level of fragmentation in the templates that we should consider? Oftentimes, if you try to cross-supply like an assay that was initially designed for a different application on something else, then the issue that people can run into is that your amplicons are oftentimes longer than the template in the sample. So for example, if you're dealing with like degraded samples, FFPE samples, your assay should be designed with that in mind, such that your amplicon length is short enough to be able to amplify like a degraded target. So I think that's an important logistical consideration that you should have. Others I would think is that the setup, for example, like, do you in future want to multiplex your assay with other targets? And in that case, like, if you know this beforehand, that down the line, that's something I would like to do. It's best to be upfront about it. Anything else you can think of, Kimi?
I just thought of like the general things that I would think most people are looking for, like specificity, and then like making sure you don't have any primer dimers so that your assay itself works and it's binding to only the target that you want.
So with that respect, I wanted to also add one more thing that generally it's a good idea to also think about what else will there be in your sample. So what's the background in your sample? Sometimes if your sample is going to be just from human, then your assay specificity just needs to be with regards to human genome. If there'll be a sample which will have a mixed community, like if it's a microbial sample, if it's going to be something for surveillance, then you have to design the assay with what you can find in the background in the sample as well.
So it sounds like, a good summary is, it's not just thinking about digital PCR itself, but about everything that's going in and your inputs and your outputs, right? But where the sample is coming from, how intact that the nucleic acid is going to be in that particular sample, and what the exact data you're looking to get. Is it multiplex? Is it simplex? Are you trying to avoid primer dimers? And is that a good kind of summary?
Yes, yeah. I like to take a step back and I like to think of the application. And I like to go from the application to the assay design and not just let's just design for this target.
Makes sense. Awesome. So you get a request in for a new assay and from somebody looking to run an experiment with digital PCR. What steps do you take to get a good understanding of what's needed and kind of how to take that from, concept to an actual assay?
The easiest type of request might come with a target sequence already included, but sometimes that's not the case. Sometimes they'll just give us like, hey, I want this mutation. So we might have to do a little research and figure out where this mutation is in the genome, get the target sequence, and then get a FASTA file for that target, and then we'll design for it. And sometimes you find, I guess, little surprises along the way. Sometimes they request a target in a region and it's like, wow, there are like pseudogenes in this region. So you kind of have to figure out how to design it so that you're not like amplifying the pseudogene, or maybe it's like in a high GC region and you have to play with parameters to make it work so that you can design an assay in this region.
Can you explain what are pseudogenes?
Yeah, so pseudogene is like a similar, or like another gene in a different part of the genome, but it has a very similar sequence to the gene that you're interested in.
Makes sense. So if you're targeting one particular gene, but other genes are very similar, you're going to get amplification of those off-target genes that you don't necessarily want to analyze. Let's talk a little bit about some of the assay optimizations that happen. So let's say you are now at the stage of you have primer and probes developed. Are there considerations after that? How do you look to see that those primers and probes are behaving as expected?
So first of all, like when we are actually designing the primers and probes, we like to see if we are avoiding any natural variation, population-wide variation, while we are designing our primers and probes. And after that, basically, we try to make sure that our primers and probes do not form primers and dimers amongst themselves and within themselves also. After that, we also like to check for specificity in the genome in terms of targets. Sometimes, if your targets are close to one another, then we need to be more creative in our designs, because we do not want competition between our probes. There, some more advanced methodologies come into play.
I was going to add, after I guess we have an assay designed and it's tested, and then we need to do some troubleshooting, like some different things we'll play with is like probe length, or like if it's like a SNP, so there's two probes, we'll play with like the way like either the probes are like right on top of each other, or we'll like stagger the start and stops. And then sometimes we'll also play with the quencher. So we have MGB or QSY quenchers, and then occasionally we'll also switch the strand of the probe, so from like the positive strand to the negative strand, just to try different ways of troubleshooting.
So I want to add a little bit to the quenchers. So we have two quenchers. One is the QSY quencher and the other one is non-fluorescent quencher, NFQ. And the probe lengths differ amongst the two quenchers. That's something that also helps helps in optimization.
Can you describe the role of a quencher when it comes to dPCR reactions?
Basically, the quencher makes it so that the fluorescent dye will not fluoresce unless the probe is bound onto the target sequence, and then when the tag comes by, it cuts off the quencher and the dye, I think, and then that's what makes the dye fluoresce.
I'm sitting over here thinking about how assay design is an art. You guys are artists. You're like the Da Vinci's of assay design.
One of our managers, he was like, "You're like a fashion designer of assays," and I was like, "I guess
sure,
so."
We can go with that. I need a female artist you're like the Georgia O'Keeffe of assay design. That's what it is. I'm in awe. Snaps to you guys.
Another question for you. When you're troubleshooting, how do you know what is the kind of tip off if a reaction is not working properly and needs some additional troubleshooting?
Generally, when we look at the 2D plots and we see how we would expect the signal to look like. There is I would say, a template of how we would like our signal to look like. We would like it sort of 90 degrees for both the dyes. When they start sort of coming closer and start making a wedge kind of a shape, and it makes it hard for us to cluster and distinguish one population from another one. That's when we start sort of troubleshooting.
Yeah, and then also specifically with SNP or like mutations, sometimes you can get the probes like I had a case recently where my wild type probe was binding to both the mutant type template and the wild type template. So then that's another case that was very obvious that we needed some troubleshooting.
It sounds like considerations for digital PCR assays are very similar to some of the considerations for qPCR assays. Is that the case? And are there any differences between designing assays for these two technologies?
Even if you design an assay for qPCR, you can try running it on digital PCR and see whether it performs well. A lot of times it will perform well, but there are some considerations for digital PCR. Digital PCR, the readout is at the endpoint, right? And in qPCR, you have like a real time readout. And so sometimes when an assay does not perform so well in qPCR, you might see that it still performs well in digital PCR because the readout is at the endpoint. Design considerations differ slightly, but overall, I do see that like the transference of an assay from qPCR to digital PCR is quite high.
So for digital PCR, does reaction efficiency play as large of a role in, like, say, it does in qPCR, in real-time PCR?
It does, right? Because you have very few templates in every chamber. So, with that respect, reaction efficiency would be a key factor in making sure that, we do get the signal that we're expecting towards the end. And since we do take pride on the fact that, we can detect low copy number uh, targets. That also makes it important that the reaction efficiency is good.
Yeah. What databases do you use? Are they public databases? And can you talk about a little bit about how these evolve and gene assignments or sequences are updated regularly?
The databases I like to use are NCBI, the UCSC Genome Browser, Ensemble, and then also we do use the Cosmic database, which is, I think, cancer mutation related.
So NCBI is basically a large repository of sequences. So anybody who's like an aspiring bioinformatician would benefit from like acquainting themselves with like NCBI because they have several databases as part of it. They have actually done a wonderful job of really cataloging all the genome sequences, but also cataloging known single point mutations or multiple point mutations or I should say multiple nucleotide mutations. And so that's a good reference point. We encourage generally our customers to give us the target information with reference to one of the standard like formats from these databases, and the ones we like are dbSNP from NCBI, where they have like really cataloged the mutations, and they also list the pathogenicity, link to a mutation or not. And it also actually one of the important or crucial metadata information that they store is the frequency of the mutations that are observed in different populations. So that really also helps us understand what's the prevalence of this point mutation across different populations. So yeah, NCBI generally for mutations and for target information and genome templates. UCSC we use or I would encourage people, like an aspiring bioinformatician, to look at it from just genome visualization and looking at if you have any tandem repeats in the genome, because they have a very nice browser where you can actually see what are the other structural variations or other components that are associated with the sequence, and they sort of overlay it very nicely. So UCSC is a good visualize. They have good visualization tool among other things.
How does bioinformatics play in? What kind of bioinformatics are you running as you design assays?
Yeah, so the design for the assay does sometimes warrant a change in our experimental setup. That's why it's very important that when we are actually developing an assay, there are two parts to the assay development. One is the wet lab, like the R&D, a molecular biologist with whom you really work closely, the bioinformatician. And so a lot of times, you might have to play with the extension temperatures. You might have to play with the extension time, different parts of the PCR, thermocycling conditions, because on the bioinformatics end, the assay design warrants longer extension time and or a lower extension temperature or something. Yeah, because we do predict sometimes that we would like a specific probe to sit before another probe. And so for playing around with all those factors, we do work closely with our molecular biologists.
And I can imagine, it can be difficult enough sometimes designing primers and probes for one target on one species, but if you throw in 2, 3, or 4 targets and multiple species, if you're looking at an environmental sample per se, and now you just exponentially increased potential pseudogenes and potential non-specific binding, and I can just imagine sometimes that the difficulties that can happen. Even when you on a computer, it looks like you have some good primers, probes, and targets identified. When you put that in an actual sample, it can cause some complicated results and interactions.
Yes. Yes, we actually launched an assay for wastewater surveillance for SARS-CoV-2, and I was on the design team for that assay. And it was interesting to see, wastewater does not have a standard definition, right? So wastewater uh, is really just a descriptive term. And so you really need to account for like very large background community. And so when we were doing the testing, you could not really take the results from one test and just predict the results on the next one as well. So that needed a fair bit of optimization, and I'm glad that it works as well it does now, but a lot of hard work went into it from in silico side and from testing side.
Yeah, makes sense. Different amounts of for lack of a better term, gunk in the sample.
Yes. Yeah.
My last question, and we'll pass you over to Cassie for Cassie's career corner. Anything else or any advice, maybe from both of you, for a scientist looking to create their own digital PCR assay in the lab? What advice would you give them?
I don't know if I just want to reiterate what Rounak has said, but definitely think about your sample type so you know your amplicon limitations. I guess if the assay doesn't do well, this is an important thing Rounak has brought up to me is yes, don't throw away the assay design just from one test not going well. I've had this happen where I was testing an assay and I did not realize that my template was RNA and I was using DNA master mix and nothing amplified. And it was not the assay's fault.
Don't worry, Kimi, I've done the same thing.
Yeah, I can reiterate on some points that I mentioned earlier, but I would like to add a topic that we didn't really touch on is the controls.
Oh, yes, controls. Yes.
It's like, when do we
You
talk
need the
about?
controls.
Yes, exactly.
Yes.
When do we talk about the controls?
That is a great point. Let's talk controls for sure.
Yeah, no. So when you're designing your assay, always first think about what's your control. So digital PCR, one of the main strengths for it is like rare target quantification, right? So a lot of times, like when customers send us SNP requests, we would assume that, oh, it might be like, a somatic mutation target or so. And so we started designing. We did a design and we tested it in the lab, and we looked at the results, and it seemed like that both, like, the wild type and the mutant were lighting up. And everyone, like, my molecular biologist counterpart, like, she came to me and she said that assay is not specific. And I said, well, the assay does, like, the results look like there might be mutant and wild type both in the sample, like, so then we took a step back and we actually looked, like, we reanalyzed the targets, and we realized that actually these are not somatic mutations. These are mutations that are just general population-wide variation targets. And the test sample, the control in which we were testing it, those controls actually had the population-wide variation. So what we were seeing was actually the ground truth. And so it's important that, like, we actually select our controls according to the target in mind. Like, so if you have like a somatic mutation, then people do end up using synthetic strings in the sample as a control, like, if or you might use like a Ceph DNA or so. And when it comes to actual, like, population wide variation, do make sure that the sample that you're using, the control that you're using, actually does not have the underlying mutations.
Taking a quick break from our conversation to tell you about Applied Biosystems Quant Studio Absolute Q dPCR system. This instrument enables quantification of your targets without the need for standard curves in only 90 minutes. Digital PCR can be as simple as preparing your samples, loading onto the plate, and running the instrument.
Unlike other digital PCR systems, the Absolute Q dPCR instrument does not use emulsion or other droplet-based methods to compartmentalize reactions. In fact, the microfluidic array plate technology enables consistent delivery of more than 20,000 microchambers. It's a great solution for anyone looking to quantify gene targets.
And Thermo Fisher Scientific has a suite of dPCR assays for applications like AAV viral titer quantification, liquid biopsy analysis, and wastewater surveillance. You can learn more at www.thermofisher.com/absoluteq or visit the Absolute Genius webpage. Again, that's www.thermofisher.com/absoluteq or visit the Absolute Genius webpage.
The Applied Biosystems Quant Studio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures. Let's get back to our conversation.
Ladies, welcome to Cassie's Career Corner. This is the part of the podcast where you don't have to think very hard. I'm going to ask you fun things about yourselves, and it's a very warm and fuzzy section, so we're here to have a good time or no time at all. You understand what I'm saying? So, we've heard a little bit about your academic journey, a little bit about your careers. How is it that each of you decided to focus in on the areas that you have? Like Kimi, for example, you said you studied pharmacological chemistry and economics. How have you decided you want to move towards bioinformatics? And Rounak, how did you decide from the get-go pretty much that you kind of wanted to be in the bioinformatics space?
So I chose basically chemistry and economics because they were both subjects that I enjoyed learning about. And then through learning more about economics, I realized that it's basically a lot of statistics, and statistics is really important in science, like regular, like chemistry, biology type science as well. So they did kind of go hand in hand, even though they don't sound like they would. And then that kind of led to me getting my graduate certificate in data mining and analysis. And because I wanted to take on a more, or I guess a less wet lab based role, I did look towards bioinformatics, and I kind of wanted to use my like interest in data analysis and statistics as well. We don't use too much of it in assay design, but I'm hoping like within bioinformatics, I can use that more.
And behold, here we are. How are you, Rounak?
Yeah, let me give you a bit of a background. I grew up in a community where everyone's a doctor.
Okay.
everyone on the street was a doctor. So it's pretty clear that I don't want to be a doctor. Yeah.
Everybody's a doctor. Okay, cool.
Okay. But that did help, like, develop my interest in medicine or infectious diseases. And I wanted to take a more research role. I did try my hands in the wet lab in the very beginning because I was exploring, like, if I would like to go towards the biotechnology side or bioinformatics side. And I pretty much realized very early on that I was an anti-talent in the wet lab.
That answered my next question, but okay, go ahead.
So I did want to build up on my strengths, and I took that as a learning, and I moved towards bioinformatics. Like in my undergraduate degree, like, it was very broad learning. We didn't really specialize in like genomics or proteomics or like, any one specific field. But as and how I started picking up my summer projects, my bachelor thesis, and I started realizing my interests really were in the infectious diseases part. I really wanted to understand, like, or rather use like, the computational tools to understand, like, how a pathogen evolves, or like, really apply it in like, improving our understanding. And I always enjoyed like working on very applied projects. So that's how I ended up like going the route of becoming like a bioinformaticians in context of like, genomics.
Okay, interesting. So, Rounak, you've had a very international like background. I mean, you studied in India, then you studied in Switzerland, and you're working like, what are all of those jumps like? I mean, that's like, those are abrupt changes.
It definitely like was a big change. I think what helped me through these different changes was like, first of all, my love for the subject. Like I really enjoyed studying. So when I landed in Switzerland, that was my first international flight.
Wow.
Actually, I had never been outside India before that. But what really helped during my master's and my PhD was, your community, your friends, your people with whom you study. Like, we had a very nice, tight knit student organization. Um, Free coffee helped.
Yes.
We could even swipe our student ID and get a free beer.
Oh,
Every
even
day.
better. Yes.
Exactly.
Wow.
I had
I
like
went to the wrong
Switzerland's
school.
where it's at. Yeah.
This was only the computer science department.
Oh, okay.
So this
Only the
wasn't the
computer
other
science
departments.
department's where it's
Yeah,
at. My bad.
exactly.
Okay.
And so like, you had like a very nice like student organization where people would hang out and like, feel like we were all in this together. You need to find like, other ways of like, sort of becoming part of like the social community there, like the society.
Right. That's great advice. I have one more question for each of you, and then I know Jordan has a fun one. My question is, and Kimi, you may have already answered this earlier, but you could give another story. But I would like for each of you, if you could, to briefly share with us your most embarrassing lab moment, and also your proudest lab moment, or like your most rewarding.
I did already give
You gave a prime
one
example.
embarrassing lab. Yes. I guess maybe another one is that on my second day of my very first job out of college. I was going into a test tube with a syringe, and I missed the tube and stabbed right into my finger.
Oh no!
Oh no!
Do you still have your finger?
Yes, I do still have my finger. It might have some tiny little remnants of the chemical that was in that syringe, but I'm still
I'm glad
alive.
you're still here. Excellent. How about your proudest moment?
My proudest moment is probably a multiplex that I'm currently working on, and it came from being given the target, and then I did all the research, found the target sequence, designed the assays, and I just on Friday tested the multiplex, and it's working.
Yes, Kimi! Woo! Celebrate!
And this is my first assay that I have like supported fully from design through to testing in the wet
Crushed
lab.
it. Well done. We're proud of you, Kimi. That's awesome. All right, Rounak, how about you? What's your most embarrassing? Because Kimi shared, you have to share. That's how it works.
I'll talk again about when I was telling you that I'm an anti-talent in the lab. So I was fresh out of high school, and I was doing like a internship in a lab, and I was supposed to pipette some samples and I think run a centrifuge or something very straightforward, like you would high school level stuff. I ended up contaminating the entire lab.
Oh,
And they had to shut it down and fumigate for like 2 days or 3 days. I don't know what.
that's juicy. Wow, we're getting all the good stuff this episode.
Oh,
And so that pretty much crossed out different options for
Process of elimination.
So, yeah, exactly. I was like, computer it is for me.
All right. And how
Yeah.
about your proudest moment?
I think working on this SARS-CoV-2 wastewater surveillance assay was a very proud moment for me. It was technically very rewarding when we sort of solved the assay design, and seeing it just applied in public health context was also extremely it felt really I felt really proud to be a part of team that contributed to something that was so relevant in current times.
That's
Yeah.
awesome. That's a good one. All right, Jordy, we only have a few minutes. Did I leave you enough time?
I think so. Yeah, this is just a straight up fun one. What is your favorite gene?
Oh, yes.
I find CYP2D6 very interesting because I'm able to use that gene in a lot of different contexts. Like I do give some seminars now and then, and whenever I need to look for a gene sequence for any application or any example I'm giving, I use CYP2D6.
I'm actually going to also say CYP2D6. Mine's a slightly more sentimental reason, though. The very first assay I ever designed that Rounak was teaching me how to design assays was for CYP2D6.
So
There you
That's
go.
awesome.
this goes to the point where I say that whenever I have to use an example sequence, I give CYP2D6.
But it was a nice challenge
Yeah.
because it had a pseudogene, so it's like it sticks out in my mind.
Awesome. Well, thank you both very much for joining us on the podcast. We loved having you. Diving into the world of assay development and your backgrounds was an honor to have you here.
Yeah, I enjoyed our conversation. Thank you.
Thank you.
That was Dr. Rounak Feigelman and Kimi Soohoo Ong from right here at Thermo Fisher Scientific. Thank you so much for joining us for today's episode of Absolute Genius. It was produced by Sarah Briganti, Matt Ferris, and Matthew Stock. With more great science around the corner in future episodes, stay curious, and we'll see you next time.
Have I told you my motto for marketing when it comes to any creative? If the response is, that's not terrible, you're golden. You never get anybody that says, that was fantastic. It's always, that's not too bad. It's not terrible.
Not bad. Yeah. We love a low bar.